Good morning. It's Miss Hunt, and today is actually Friday, the 6th, and I wanted to go over the lesson that I taught yesterday. So anybody who was absent or my online learners can have a chance to go through it. Yesterday, you were going to do this worksheet, okay? And we were also going to take that quiz that was on the lesson plan for yesterday. So I added a little bit of DNA review into there. So hopefully you at home have some kind of model of DNA. Either you got this from the worksheet or you have drawn it out on paper. So I wanted to go through it one more time with you. First of all, we have to remember that these are a nucleotide. We have a sugar, a phosphate, and a nitrogenous base. Ooh, there we go. And these three things right here are connected by the nitrogenous base by a hydrogen bond between here. Hopefully you remember the base pair rules of Chargaff's base pair rules. C binds with G and A binds with T. Okay, so we started off class yesterday. We had a bunch of construction paper, and I asked everybody to make something to represent the 4 enzymes for DNA replication. So here's your enzymes. We have helicase, RNA primase, RNA polymerase, and then ligase. So we decided that we would pick any colors we wanted to and draw something that would help us visualize this process. So here are mine, and yours doesn't have to look like mine, but it helps to add emotion into our learning. So this is a kinesthetic learning. And if you act it out, you'll probably learn it a little bit better. So this was my first one, helicase, that first enzyme. I put it in red because it was getting everything started. It was going to cut up the DNA. It kind of reminded me of like cutting open something. It was bloody. I don't know. It made sense in my brain. The scissors I put there because it cuts through the DNA and breaks those hydrogen bonds. So that's why I chose red and a pair of scissors on my helicase. The second one, the RNA primase. This is going to lay down the RNA code to start so that DNA knows where to begin replicating. I made it green as in go. I put a start flag on it so I could say, this is where I start off my DNA replication. The third guy, DNA polymerase. I put him as kind of having a construction hat on. It may or may not look like a hat, but I put construction zone. I chose orange because it's the color of construction, because this guy's going to be building the nucleotides. And our last one, ligase. Ligase, I put yellow because it reminded me of glue. I put a little glue bottle on there. And I put OK because it glues the Okazaki fragments on the lagging side. So I kind of put that up for me to remember. Okay. I also put up on the board this story right here. And let me make it a little bit bigger here. Okay. So I kind of put it up here for the story of DNA replication, okay? So you can say once upon a time, but here is our DNA replication. Helicase is going to come in first and unzip the DNA. RNA primase lays down the RNA code as a start position. Remember RNA code, there's 2 things. 1, we use uracil instead of thymine. And 2, we use ribose sugar instead of deoxyribose sugar. Then the DNA polymerase builds nucleotides. Remember the 3 parts to a nucleotide: a sugar, a phosphate, and a nitrogenous base. He works towards the 3' end. Remember, the leading side is going in the direction of the 3', and that's very easy. The lagging side, he has to work backwards in order to go through it, and that gives us the Okazaki fragments. Finally, the ligase is going to glue the fragments together. And we end up with 2 new strands of semi-conservative replication. Remember, that means half of it's new and half's old. Remember, it does this so that it doesn't have mistakes. It has something to base the new one off of. Otherwise, we copy from the old and then we throw it away and we just get the new one. We're more likely to have a mistake left over. Okay, so after the kids read through this and we made our enzyme models, then we took our DNA out, okay? And I had the kids explain the story using their enzymes to a partner in class. So we had to take a pair of scissors, and the kid said, okay, I'm cutting open the DNA. This is what the helicase does. Ooh, there we go. He unzips the DNA. Okay, and they could look at the board throughout the period to practice it. After each kid told the story to their partner and their partner told the story to them, I called on kids by name and had them explain it to me. And we hopefully had some good learning go on during that process. So I'm going to model what the kids did to each other. So again, I said that helicase unzips the DNA. The RNA primase comes in, and I'm going to flip open my DNA here. Okay. And he's going to lay down an RNA code as a primer, as a start position. He lays it down, RNA code, which means uracil instead of thiamine and ribose instead of deoxyribose. Okay. Then the DNA polymerase comes in and he starts to build the nucleotides. Now, if you have extra nucleotides, then you could actually lay this down on paper and kind of build it. There goes the DNA polymerase building. Trying to hold this all in my hands at the same time is kind of hard. There we go. Got the DNA polymerase and he builds this guy on. Okay, good. There we go. And then this is a C and he would bind with the G. So he could build that on right there. Those are my sound effects for building. Okay. All right. He builds really easy on the leading side because it's going in the 3' direction. On the lagging side, he's got to work backwards, which means he goes up. Oh, let me throw this part up. Okay. Which means he would go up and then back down. So we'll start here. He'll go here, up, and then he'll move back down, and up, and then he'll move back down. This leads us to the last part, the ligase. He's going to glue the Okazaki fragments together, and we're left with 2 new strands of DNA with semi-conservative replication, half old and half new. Okay, practice saying that and practice reading the story and going back over that. All right, then you're going to do the DNA worksheet. Okay, all right. When you get here, the beginning part, this is pretty easy. Okay, the picture was a little bit challenging because it laid down 3 parts and it didn't explain it. So you have to reword it in your own words. I would try to explain the 3 enzymes. So here in A, I would definitely talk about helicase. In B, you could be talking about that the RNA primase primes it and the DNA polymerase builds it. And then in C, you could be talking about the ligase and about it being 2 new strands of semi-conservative replication. Okay, but rewrite it in your own words because that makes you learn it better. Okay, all right, the rest of the questions should be pretty easy. On the back, remember here you're just adding the complementary pair. So for this A, you would put a T, and for the T, you put an A. On this one the kids got a little confused on. So let me show you mine. You have to fill out the box. So the A binds with the T, and the T binds with an A, and the G binds with a C. And then you take the black side right here and you rewrite it. Rewrite it right here. And then you make the new base pair off of that one. So it's almost like I'm splitting this part up. The TAC in blue, I rewrote it right here, TAC, and then I put the base pair. So I'm putting 2 new strands from my original strand. So you could call that the parent strand, and then I'm making 2 strands off of that, okay? At some point today, I will do a Google Meet in case you have any questions about anything. Hopefully you're able to do the quiz today on the Google Classroom. It's on a Google form. And we are actually going to finish DNA next week. I was hoping to have DNA finished this week, but we need a little bit more time. So we're going to finish DNA next week. And I hope you guys have a great weekend.